She's been a member of the faculty here since 2006. I'm happy to call her my colleague, and I'm very thankful that she is willing to share her expertise with us tonight. Just a couple of logistics, if you can keep yourself muted, that would be really appreciated. And if you have questions, you can put them in the chat, and I will be moderating the chat. The chat will be coming directly to me, and I will be asking the questions for folks. So without further ado, I present to you Dr. Jennifer Brigati.
Hi, thank you so much for that introduction. Really appreciate it. So tonight I'm going to be talking to everyone about the COVID vaccine, and I'm super excited to get a chance to talk to you all about this. To me, it really does represent hope in a syringe. Hope that the next time I give a presentation, it will be to people sitting in an auditorium instead of sitting in my office by myself. So I do want to really just give everyone as much information as possible. It has made this a little dense with all the questions that I got, but I'm going to try to get through it in about 40 minutes so that there'll be time for additional questions at the end. All right, so what am I going to talk about tonight? I'm going to start with some basics of how the immune system works, because I know not everyone watching this is likely to have had any sort of immunology type course. And then I'm going to talk specifically about the messenger RNA vaccines. Those are the two that already have emergency use authorization. And I'm going to talk about the history and development of those. And then I'm going to talk about the adenovirus-based vaccines. One of those is actually up for FDA evaluation tomorrow, so we'll be talking about that and talk a little bit about their history and development as well. I'm going to talk about the clinical trials process in general and how that has been accelerated to work for these COVID vaccines, and talk a little bit about the safety and efficacy data that has come out so far for these vaccines, as well as the common side effects, and then kind of some time for Q&A, answering some of the questions that I got ahead of time that I couldn't really fit in anywhere else in the presentation, and then also taking whatever questions get sent to my colleague, Dr. Siopsis, during this presentation. So to start out with some immune system basics, there are two broad branches to your immune system. You have your innate immune system and your adaptive immune system, and they work together. So your innate immune system is the things that happen very quickly. And these are kind of the same no matter what the invader is. You know, any virus is going to get a certain response. Any bacterium is going to get a certain response. So they're not terribly specific. They recognize that it's a virus, but not what virus it is. And these parts of your immune system work the same, whether it's the first time you're seeing the virus or the 50th time you've seen that virus. The next part of your immune system is the adaptive immune system. And these two parts do work together, the innate and the adaptive systems, but the adaptive system is much slower on its initial response. And so your initial response with adaptive immunity is going to take somewhere from 7 to 10 days to really get any sort of effective response. And your adaptive immune response is specific for individual invaders. And we say it is specific for something we call antigens. And the word antigen originally came from antigen generator or antibody generator. And so the idea was that these are things that your immune system can recognize. And so they're little parts of viruses, little parts of bacteria that your immune system can specifically recognize, but it's not going to respond well to them the first time it sees them. And so you have to see it once and be activated, and then you develop memory. And so your adaptive immune system works much better the second and subsequent times that you see things. Now, there are two branches to your adaptive immune response. You have cell-mediated immunity, and cell-mediated immunity is things like your killer T cells that can get rid of virally infected cells. And then you have humoral or antibody-mediated immunity. And to have a really effective response against a virus, you need to activate both of these. And so these are all really important, and you can't get by with just innate immunity or just adaptive immunity. You need to have both. And so what happens when you have any invader come in is that your invader has in it both some broad categories of things that you're likely to recognize, and those will be recognized by your innate immune system and will trigger a reaction that includes things like non-specific cells, like macrophages that you have that will attack any invader and eat them, and then some chemicals that you'll release in response to that. And then those things will trigger your adaptive immune system. And your adaptive immune system recognizes these antigens, these specific little parts of the invader. And your adaptive immune system is dependent on the innate immune system. And so it's important to remember that an effective vaccine needs to stimulate both of these things. So this is a really busy slide, and we're not going to spend a lot of time looking at the details of it, but I wanted to show you what we think the actual normal response is to SARS-CoV-2 in your body. So what happens when you get infected with this virus naturally? And let's just get rid of everything that's going on inside that cell, because that's all has to do with gene expression and is not really, that'd be a whole nother talk. So for now, let's just focus on what happens, what the outcomes are. So the virus infects the cell, and the cell immediately sends out some emergency signals like, I'm infected, there's a problem here. All right, so that's what you're seeing down here. So this is an innate immune response. That includes these things called interferons. Interferons are a signal that you have a viral infection. They tell other cells to put up their defenses. And they also are part of what make you feel bad when you have a viral infection or when you get a vaccine that would trigger their production. So that's something to keep in mind for later. These are all important in triggering the adaptive immune response that they're showing over here. So this macrophage is also part of your innate immune system, and it is able to take in the antigens, the little pieces of the virus, and display them so that your immune system can recognize them, because some parts of your immune system can't recognize just that virus floating around out there. And so the macrophage will display it and say, hey, T cell, come over here. And so here we have a T cell that is now recognizing that antigen. And this could be a T cell that will become a helper T cell that will help with the antibody response, or it could be a T cell that will become a killer T cell that can actually go and kill virally infected cells. You'll also have an antibody response. I think they have a B cell around here somewhere. Here's the B cell. And so the B cell will recognize this virus directly, but we'll also get some help from this helper T cell down here, and it will produce antibodies. So you can see that there's a lot of parts to that normal host immune response to SARS-CoV-2. And so if we want a vaccine to work, we need to have a vaccine that will be able to make all those things happen, but happen without you getting sick from the virus. Now, I did get a question that had to do with whether or not vaccines stimulate better or longer lasting immunity than real infection. And so this is a look at the kind of immunity you'd expect to have stimulated from a real infection. And the thing about a real infection is that when we talk about antigens, these little pieces of proteins that our immune system is responding to, this virus has lots of them. So there are actually about 12 different proteins that are expressed from the SARS-CoV-2 genome. And so you could have responses in a natural infection to any of those proteins. But it's really just 2 of those proteins that are helpful when it comes to making it so that you don't get a repeat infection. And those are for the N protein, which is basically like the outer coat of the virus, and then the S protein, which is that spike protein, and that's the one that all the vaccines are targeting. So the reason why you might have better immunity from a vaccine is that you're having an immune response then that's only targeted toward the parts of the virus that your immune system needs to recognize to prevent reinfection, rather than potentially responding to all sorts of things. Now, that's not to say that for every disease that the vaccine is better at preventing reinfection than the actual disease. That's not always the case. But in this case, there is some preliminary data for the Moderna vaccine that indicated a greater production of neutralizing antibodies in folks who were vaccinated than in those who were convalescing from actual COVID-19 infection. So it does look like this may be one of those diseases where you will get a better immune response from the vaccine than you do from the actual virus. So let's take a look at how some of these vaccines actually work. We're going to start with the mRNA vaccines. Those are the 2 that currently have emergency use authorization from the FDA. And if you've ever looked for information for these and found that you couldn't find much, might be because the names have changed. So what we call the Pfizer vaccine, if you're ever looking for information for this, it's really called BNT162b2. I know that just rolls right off the tongue, right? But if you're looking for scientific data on it, that's what you have to search for. And then for Moderna, it's mRNA-1273. So those are the names that if you're looking for clinical trial results or something and you really want to dig into the data, that's what you would have to search for. So how do these work? I really like this picture that was made by the American Chemical Society. It really walks you through it nicely. So we're going to start right here. Here's our messenger RNA vaccine. And all this has in it is something that we call messenger RNA. Well, what is that? It's been a while since you've had any sort of biology or genetics class. You probably don't remember what the different types of nucleic acids are. Well, DNA is sort of the storage molecule for all the information in a cell. Messenger RNA is just a carrier molecule. It doesn't last very long. It's very unstable. Now, with viruses, a lot of viruses are just made up of RNA. And actually, in this particular case, the virus that we're trying to protect against is just made up of RNA. So it makes sense that we might try a messenger RNA vaccine for this particular virus. And so this messenger RNA vaccine, when it comes toward your cell, it fuses with your cell membrane and releases the messenger RNA. And so notice that it's not ever going into the nucleus. It's not incorporating into your DNA or anything like that. It's entirely staying in the outer part of your cell, the cytosol. And it just gets translated by your ribosomes to make the spike protein that was found on the virus. So you're not being infected with the virus. You're really just getting instructions for how to make the spike protein, and that's it. Once that spike protein is made, it can go a couple different places. One is that it can be displayed on the surface of the cell or released from the cell, and B cells can recognize it that way. Another place that it can go is it can get chewed up, which happens to all proteins that are inside your cell. They get chewed up, and it will get displayed on a special type of molecule you have that moves to the surface of the cell, and that will hold that antigen up, that little protein up, so that your T cells can recognize it, all right? So this is one type of T cell, and then there's a slightly different type of molecule that will hold it up for the other type of T cell, for your helper T cells, all right? So that's the gist of what these vaccines are doing. They're just basically allowing your cells to produce a viral protein the same way that it would if you were infected with a virus, but you're not infected with a virus. And so it reduces that risk associated with actually having an infection, but it still triggers both a cell-mediated and an antibody-mediated immune response. So you'll get a really strong immune response from this. And then one of the things that's nice about this is folks really worry, like, is this changing my cells? Well, messenger RNA is really unstable. I often have my students do experiments where they isolate messenger RNA, and I talk to them about how quickly we have to work, or it will just totally disappear. So if you've heard about vaccine spoilage being a big issue and that these have to be stored at really cold temperatures, that's because that messenger RNA is highly unstable and it's not going to stick around in your cell for very long at all. So you don't have to worry about it long-term doing anything to your cells because it's very unstable. So that messenger RNA goes away, and then your body recycles things a lot. And so these proteins won't hang around either. The proteins, once that messenger RNA is no longer there, you're not producing more of that protein will go away. And we also recycle all these little things that present antigens to other cells. We recycle those as well. So they won't be presenting those antigens anymore over time. And if anything did happen, and for some reason the cell did continue to present those antigens for longer than we expect, then one of your now activated killer T cells would come along and get rid of this cell. And so you really wouldn't have any issues with this being a long-lasting change. And so all you're really left with at the end of this is that you have your killer T cells that are cells that can get rid of virally infected cells. And then you have your antibody producing B cells over here. And you'll have memory cells. And so you are just left with this immune response and not a whole lot else. Okay, so that sounds great, but how can we know that these are really okay when they're so brand new? Well, they're not really brand new. And so it's funny because I started teaching a long time ago, about 14 years ago. And when I did, they were talking about these types of vaccines in the textbooks. And so for years, I've been like, okay, when are we actually going to see a vaccine like this? Because these are not a new concept at all. It's just that they haven't really been used. They were kind of waiting in the wings for when we had an opportunity to use them. So this paper from 1989, which I hate to think about this, but that was 32 years ago, is one of the first papers where they really looked at how can we put messenger RNA into a cell. And then here's a paper from 1993, which is 28 years ago, and so which longer than I want to think. But this was the first trial in mice where they showed that you could create a messenger RNA vaccine, and this one was for influenza. And so this is not a new technology. It's something that has been developed for a long time. And then this is a review article I found in 2012 that was just talking on and on about how these were such a great new way that we have got to get these technologies out and start using these technologies. And that again was 9 years ago. So lots of background on these messenger RNA vaccines. And then I really like this this little timeline that shows, this is kind of our background research and discovery work very long time ago. But then here's where the R&D happened. So a lot of times when we look at how long it takes to develop a vaccine, we're thinking about the time that it took for all the research and development. Well, that research and development occurred over a very, very, very long time before we got to 2020, where we started looking at this for the COVID vaccine. And in fact, the COVID vaccine is not the first one to go into clinical trials for these messenger RNA-based vaccines. There are more than 30 trials that have already been completed, actually, and many more, around 100 more that are underway. And many of those were started before COVID-19. So there have been a lot of cancer therapeutics that were based on messenger RNA vaccines, as well as a vaccine for rabies, respiratory syncytial virus, Zika virus. And this was the earliest one, was in 2008, phase 1 clinical trial results for a messenger RNA vaccine. And I got a question about long-term side effects. And so I just want to kind of point out that, this particular trial was about 13 years ago. And so we would expect that if there were going to be some unusual long-term side effects, that we would have seen those in folks that had participated in the trials like this. I had also gotten a question that had to do with the ingredients that were in these, and asking which ingredients in the messenger RNA vaccines are new and which ones have been used before. So I made this slide with a little bit of color coding for you. And so the ones that are in dark green are found in other FDA approved vaccines. And the ones that are in light green are found in other FDA approved injectable drugs. And so that's a lot of the ingredients are in those colors. And then the ones that I put in blue are in other vaccines that are in clinical trials. And so that includes most of the stuff that's up there. There are a few things in here that obviously are going to vary from vaccine to vaccine. So exactly what the messenger RNA is coding for is going to vary. We wouldn't expect that to change the safety profile, but that is going to be different with each vaccine. The lipids are a little bit different. There have been lots of different drugs that use lipids, but these particular lipids are a little bit different for some of them. The Moderna vaccine, there may actually be others in clinical trials that have the exact same components, but that information is not fully released. But since they do have other similar vaccines in clinical trials, there's a good chance that they're going to have using the same lipids in those vaccines. The only thing in the Moderna one that was a little bit different that I didn't find in another injectable or vaccine was acetic acid. That surprised me because acetic acid is something that's used in the lab a lot. And there's a lot of other acids that are used. These are again just used to adjust the pH of the vaccine. There's no reason to think that acetic acid would cause any problems in the cell, but it was one that I did not find in another injectable. It is in a lot of drugs, though, that the FDA has approved. So that was what I found as far as answering which ingredients in these vaccines were new versus which ingredients had been used before. So how do these messenger RNA vaccines compare to other FDA-approved vaccines? So there's a lot of different types of FDA-approved vaccines, but since I had to fit this onto one little slide, I kind of condensed them into two broad categories. So I have over here our live attenuated vaccines. And those are vaccines that I'm sure you're familiar with, things like the measles vaccine, chickenpox vaccine, those are live attenuated. And so basically they're a viral strain that just doesn't replicate quite as well in humans as it used to. And then we also have other types of vaccines that are called either inactivated vaccines, or there are also, so they can be the whole agent, like the whole bacterium or something that's been inactivated, or subunit vaccines, so just a particular part is being manufactured and then that's being injected, or toxoid vaccine. So that would be like for the tetanus toxin, where they've just made it so that the toxin is not going to cause illness. So these are vaccines where there's nothing living, all right? And so then I'm comparing, so we've got living vaccines, non-living vaccines, and then we've got our messenger RNA vaccines. All right, so how are they the same and how are they different? So they can all stimulate antibody-mediated immunity quite well. The big difference is in their ability to stimulate cell-mediated immunity. And so live attenuated vaccines do that very well. And messenger RNA vaccines do that very well. But these inactivated and subunit vaccines, and toxoid vaccines, don't do that very well. Because in order to stimulate cell-mediated immunity, you have to have your immune system think that whatever it is that was infecting you or that you're producing came from inside of your cell, because that's what viruses do. Viruses make stuff inside your cells. And so some of the traditional vaccines just really can't trick your immune system that way. And so they don't work out as well for cell-mediated immunity. And cell-mediated immunity is important for fighting off a virus. Now, when it comes to needing booster shots, there's a little bit of a question mark for the messenger RNA vaccines, and I had gotten a question about that. How long will the immunity from this last? We have a little bit of information on that, but not much, of course, because this is a relatively new vaccine. Typically, when you have a live attenuated vaccine, you don't need regular booster shots. You might need 2 to start with or 3 to start with, but then you usually then have lifelong immunity. With inactivated vaccines, you typically do need booster shots. And the messenger RNA vaccines, we think could be somewhere in between this, but only time will tell for that. There have been a study where they looked about 4 months out, and they have found that there's only been a small decrease in the amount of antibody being produced in folks that were vaccinated. So that's pretty encouraging that we'll continue to have immunity for some time. All right, I also had a question about safety in pregnant
Dr.
women.
Brigati, can I just interrupt you for 1 second?
Yes.
It's related to what you were just talking about. Can you explain one more time, what does possibility of reversion to virulent form mean?
Yes, I haven't gotten to that one yet, but I will for sure.
All right, sorry, I'm monitoring chat and listening, so sorry.
Okay. Yeah, that's fine. I will get to that one right after they're pregnant because I'm kind of moving down the aisle here.
Sorry.
No, you're fine. Okay. So the safety for pregnant women. Generally live vaccines are not safe for pregnant women, but inactivated vaccines are. Messenger RNA vaccines, we do not have that data yet. Those clinical trials have not been completed. There are some starting, but we don't have that data yet. So at this point, what pregnant women are doing is they're consulting with their doctor and weighing kind of their personal risks. So do they have a lot of risk of developing COVID-19? For example, if they're a healthcare worker, maybe then in that case, it is worth it to get vaccinated at this point. If they're in a lower risk situation where they're not going out a whole lot, then maybe they want to wait and not be vaccinated at this time. So we don't have that data just yet to know for sure how safe these are in pregnant women. There's no reason to think they're unsafe, but we don't have the data yet. The same thing for folks who are immune compromised. Again, typically you're live attenuated or not safe in that population, your inactivated are. We think that these will be safe in folks who are immune compromised, but we don't have that data just yet. Now, this next one about possibility of reversion to a virulent form. So with traditional live attenuated vaccines, the way that those are produced is just by culturing the virus in something it doesn't really like to grow in. So typically when you culture viruses, you culture them in cells, human cells. And so maybe you culture them at a different temperature, or maybe you culture them in cells for a primate or something, so that they make some adaptations so that when you put them back into human cells, they just don't grow quite as well there. When you do it that way, which is the traditional way that live attenuated vaccines have been made, there's no guarantee that those viruses can't mutate back to what they were. Now, we have not had that happen for very many vaccines in history. In fact, the only one that I know of that has ever happened with is the oral polio vaccine. The oral polio vaccine is a very effective vaccine, and that's why it's been still used in some places, but they're really phasing it out now. We don't use it in the US because that oral polio vaccine contains a strain of the polio virus that is attenuated but can revert back to wild type and cause polio. And so we don't use that in the US anymore. Because of that, we use an inactivated vaccine that is completely safe. But this is always a concern if you use a live attenuated vaccine. And that's why the messenger RNA vaccines, one of the reasons why we think they're going to be much safer than these live attenuated vaccines. Maria, does that answer the question that we had?
Yes, thanks. Sorry about that.
Oh, it's fine. Thank you. All right. And then one question I've gotten from folks in the community is, does this make your cells express viral proteins? Well, yes, because it has to, because a virus makes your cell express viral proteins. And so any sort of a live attenuated vaccine is going to do that as well. And that's part of how it works, is that your cells will transiently, for temporary time, express those viral proteins so that your immune system will appropriately attack them like they came from a virus. So they do express viral proteins because they have to for it to work, but it's not a long-term thing. It's not any sort of a permanent change in your cells, and it's going to go away very quickly. So it's not something that's of any safety concern. All right, now to move on and talk a little bit about the other two vaccines that are coming up soon, one of which is up for possible approval maybe tomorrow or early next week. They're going to start talking about it tomorrow at the FDA. So these are what we call replication deficient adenovirus vector-based vaccines, and that is a mouthful. And so these vaccines are based on a virus that has been studied for a long time, and I'll go into the history here in just a minute. But we know a lot about these adenoviruses and different strains of adenovirus and how to work with them. And so these particular strains have been made so that they can't multiply in your cells. So they really wind up working almost the same way as the messenger RNA vaccines, because really all they're doing is delivering some genetic material to your cells so that you can make that same spike protein that's being encoded in the messenger RNA vaccine. So the Oxford AstraZeneca vaccine, which again, if you ever want to look up information, it's not going to be called that. It's going to be called ChAdOx1, is how you would find that one. It is done using a chimpanzee adenovirus. So this isn't even a human adenovirus. It's one from chimpanzees. And it's been modified so that it can't cause disease and it can't replicate in your cells. And then they have taken that spike protein and they have put that into, well, not the protein, they should put the gene for the spike protein into the genome for this ChAdOx1 virus. And then you get injected with that, and it can't cause true infection because it can't replicate in your cells. So it really just enters into your cells like the mRNA vaccine. For the Janssen or Johnson & Johnson vaccine trial, they are using a human adenovirus. It's human adenovirus 26, and it has also been modified so that it can't cause disease. It is replication deficient. It can't replicate. And again, they have taken the gene for that spike protein, put that into the genome for this Ad26 virus, and that is what you are injected with. Now, these adenovirus-based vaccines, again, work very similar to the way that the messenger RNA ones do, but a little bit different because an adenovirus is actually very different from the kind of virus that SARS-CoV-2 is. So it's very different from coronaviruses. So adenoviruses are DNA viruses. And so in this case, that copy is within the DNA of this virus. So when you get injected with this vaccine and that adenovirus makes its way into your cells, it is releasing its DNA into your cells. However, Ad26 is an adenovirus that does not integrate into host chromosomes. So it's not going to become a permanent part of your DNA. It's just in there temporarily, just like the messenger RNA from the last set of vaccines. But it's going to do the same basic things. It's going to be able to be, you'll turn it into messenger RNA, you'll make the spike protein that will be presented in multiple ways to your immune system so that you can trigger both cell-mediated and antibody-mediated immunity. So it really, once it's in the cell, the process looks just like it did for the messenger RNA vaccines. These vectors don't persist in the body for long at all. In fact, a challenge to using adenovirus vectors is that sometimes your immune system gets rid of them too quickly. Ad26 is one that's not super common, so most folks have not been naturally infected with that adenovirus, and so hopefully their immune systems will not just instantly get rid of it, so that's why it is a reasonable thing to use for a vaccine, but these definitely are not going to hang out in your body for long. So what's the history behind these? So you can see this is a paper from 1996, which again is longer ago than I'd like to think. And so this is one of the early papers showing that you can use a replication deficient human adenovirus as a vaccine carrier. And so again, this is something that has been in development for a very long time. And then here was a nice review from 2004, just talking about all the different ones that are in testing, from malaria to HIV, all the different types of vaccines that are being tested using adenovirus-based vectors. Now, adenoviruses have been used for a very long time. And so some folks, I think I got some questions, or maybe I just have heard in the community some questions about, is this like gene therapy? Well, I can understand where that question comes from, because there has been use of adenovirus vectors in gene therapy. It turns out they don't work super well for a lot of things, and that's why they actually are going to work okay for a vaccine. They just don't persist super long. So that's been the challenge with gene therapy. But clinical trials with adenoviral vectors began in 1993, and originally they were using ones that were not replication deficient, so they wanted these to replicate because they wanted them to hang around. And this picture over here is from the very first gene therapy clinical trial in 1993, the first patient that was infected with a recombinant virus. And so there was varying success with this, but there were also some issues. There were some safety and efficacy issues. And so that meant that there had to be a lot of changes made to those viral vectors. And that is what makes me confident that these ones are safe, is because we have had a whole lot of research done and have found which ones are not so safe and which ones don't work well. And now we have moved on to these replication deficient ones, which are much safer. And so there have been quite a few clinical trials using these replication deficient adenovirus vector-based vaccines, particularly this one Ad26 that is in the Johnson & Johnson vaccine that is going to be going up for emergency use authorization potentially tomorrow. So it has been, you might have even heard of one of these. So Ad26-Zevo is one for Ebola virus. And that has been used in a large clinical trial. And also it got the equivalent of EUA in Rwanda because, of course, Ebola virus is such a huge problem in some areas. And so they did a government-sponsored vaccine campaign with that there. But they also have done trials for this for HIV, malaria, RSV, filovirus, Zika virus, and HPV. So lots of different ones have been tried. And this one has been around since 2013 using this adenovirus 26 replication deficient vector. So plenty of clinical trials on this, and so we feel pretty comfortable that this is working out pretty well. Johnson & Johnson has that Ebola vaccine, and it has actually been used in over 100,000 people already, and has had a good safety record. So we feel pretty good about that. All right, so how do these compare to these prior FDA approved vaccines without spending too much time, because we've already talked, these two columns are the exact same as what we had before. So how do these compare? They're very similar to the messenger RNA vaccines. So they do stimulate antibody-mediated immunity and cell-mediated immunity very well. We don't know if these are going to require booster shots, but we think there's some good possibility that they won't. So this Johnson & Johnson shot that is going up tomorrow to the FDA is one that is a single shot, a single injection. And so hopefully this is one where we can get away without the booster shots. Safety for pregnant women, there's a little bit more data here because of that Ebola vaccine. And that Ebola vaccine has not caused problems with pregnancy. Again, we don't have controlled clinical trials for this just yet, so I would still classify this as to be determined, definitely something to discuss with a healthcare provider. But there's maybe a little bit more data on this one because of that Ebola vaccine. And again, we don't expect there to be big differences based on whether it's making a spike protein for SARS-CoV-2 or a protein for Ebola. So it should be pretty similar. Same thing with safety for immune compromised individuals should be similar to what we've seen with the Ebola vaccine. And no issues with reversion to a virulent form. But again, yes, of course, your cells are going to be expressing those viral proteins because that's how the vaccine works. All right, so now to talk about this clinical trial and FDA approval process, because that's, I think, what makes people the most uncomfortable, is that it seems like this went really, really fast. And some parts of it did, But I think the parts that went the fastest are the parts that you usually don't ever see anyway. So this part here, I really like this little diagram, even though it's more for veterinary medicines than anything else. Um, this part here, the research part, and then the part where you look at animal models, that's usually the really, really, really long part. So that could be 3 years, that could be 5 years, that could be 10 years, that could be really long. That was really sped up here because all of these vaccine candidates had been in research and development for 20 plus years. And so they had done that basic science research. These were plug and play. Like all you had to do was switch out what gene was in there, what spike protein you're encoding, and they were ready to go. So it was very easy to breeze through that initial part. Animal models, they did still do some animal model studies before they were approved for clinical trials, but they also could use preclinical data from related vaccines. So ones like for the Ebola virus or for Zika virus. And so that allowed them to get through these phases pretty easily. And then they apply for the investigational new drug status. And so that application has to include that animal pharmacology and toxicology info. Has it ever been used in humans, or has anything like it ever been used in humans? And then how do you manufacture it, and how are we going to study this in the clinic? And then if that gets approved, then you move on to your clinical trials. And I really like this graphic from Nature that explains how these clinical trials got to go a whole lot faster than normal. So again, normally this part over here where your preclinical trials can last a really long time, and that really didn't take very long at all for the SARS-CoV-2 vaccines because that stuff had already been done, and so they just had to switch out the spike protein for SARS-CoV-2. And I keep saying the spike protein, but they did test a fair number of different proteins. I mean, there were only 12 to test, but they did test a variety of proteins to see which one would induce the best immune response in animals. They didn't just skip straight to the spike protein. So this was fast. So once you got past this investigational new drug, then how did we get through the clinical trial so fast? Well, a lot of times you run your phases separately, phase 1 and then phase 2 and then phase 3. Sometimes they combine them, but a lot of times they're separate. And those phases can take a long time because you have to get people to enroll. And generally speaking, it's usually hard to get people to enroll for clinical trials. And so, one of the big places where both of these technologies have been used is for cancer vaccines. And you can only, like in phase 1, typically it has to be someone who has exhausted every other option for cancer treatment before you can enroll them in your trial. And so it can take a long time just to get people enrolled. And so that's why these phases oftentimes take a really long time. And then you typically will want to have a full analysis of the data from each phase before you move on to the next phase. In this case, they were able to, first of all, quickly enroll people because folks were all anxious to have some way to get past this pandemic. So quickly enroll people into phase 1 and overlap the phases because we had some safety data for these vaccines in the past. Phase 1, which is really focused on safety, they were able to accelerate that. And so phase 1 and phase 1 and 2, they focus on safety and on dosing. And then once it was clear that, okay, this is relatively safe, we don't have any real concerns, then they move straight into phase 3. Phase 3 is where you enroll a lot of participants. In the case of these vaccines, 30 to 40,000. And it was easy to get those participants for this because everybody wanted to be protected from SARS-CoV-2. No one wanted to get COVID. And so it was easy to get those folks enrolled very quickly. They used many different sites for their clinical studies. And so that allowed them to be able to run through this process quickly. What also helped is that the endpoint that we were looking for was just whether or not someone got COVID-19, or in some cases, if they got severe COVID-19. And so since we had a really bad outbreak, obviously going on, it made it so that we got to those outcomes very quickly. So we got folks in the control group that got sick very quickly. And so that allowed us to quickly be able to evaluate how well the vaccines were working. So there was some amount of relaxation in, allowing folks to move through the phases a little quicker than they normally would. But the amount of data that was collected was still the same as the standard. So there were no corners cut there. Another thing that happened is that normally you don't start producing whatever it is you're trying to get, approval for until it's approved. But in this case, we had a lot of companies that were starting production before they had approval, because there was at least some realistic understanding that there was a good chance that they would get approval and they would be able to move through and make money off of this. But that was at risk. Whereas normally most companies are not willing to take that risk. They're not going to start production until after they have approval. Now, it's important to recognize that what they have right now is not FDA approval. They have emergency use authorization. And so this thing right here where it says BLA submitted, that's a biologics license application. Typically, you submit that and you've got a review time of 1 to 2 years. All right. Right now, they're speeding up their review by a lot. And so this review process is really just taking a month or two. All right. So it's a much faster process because we're not getting full FDA approval. We're getting emergency use authorization. So what is emergency use authorization? Emergency use authorization is just where the FDA commissioner is able to say that we're in a desperate situation. We have a public health emergency. There's no really adequate approved way to treat this, to fight this. And so we can approve the use of this thing for this emergency to diagnose, treat, or prevent. For a vaccine, the application for emergency use authorization, it has to include all the data from phase 1 and phase 2. So that's your safety data. And then phase 3 is your main efficacy data. And so you have to have that with a median follow-up of at least 2 months, completing the full vaccination regimen. And then in addition, you have to have additional safety data, not just from phase 1 and phase 2, but also for at least 3,000 of the vaccine recipients in phase 3. And honestly, in the publications that I have seen coming off the mRNA vaccines, that has included safety data for all of the phase 3 participants. Now, emergency use authorization is not FDA approval and it can be withdrawn. And so that's important to remember as well. So they continue to tightly monitor safety and efficacy as they go through these continued use, because it's not exactly a clinical trial, but they want to know exactly what's going on and how well it's working. And so there have already been some emergency use authorizations that were revoked with regard to SARS-CoV-2. And so they had originally given an EUA to using chloroquine and hydrochloroquine for treatment. And then they found that that wasn't helping. So they revoked that EUA. All right, so if it's possible for it to get revoked, how would that happen? So how do we monitor that safety? So there are several ways. Most folks are familiar with VAERS, that's the Vaccine Adverse Event Reporting System. This is a spontaneous public reporting system, and it's a way where anyone can go on and just report that they had an adverse reaction to a vaccine. And so the raw numbers from this don't mean a whole lot. It just means that something happened to someone around the same time they had a vaccine, but the CDC investigates anything that is significant and severe that's reported on here. And especially if there's any deaths reported, the CDC investigates that. For the COVID vaccine particularly, they have a really neat thing called vSafe. And so if you get the COVID vaccine, you may be asked if you would be willing to join this. And it's an active surveillance system. And basically they send you some texts and they ask you each day, like, what kind of symptoms are you having as a response to this? And have you had to call in sick to work? Did you have to seek any medical care? Did you have to take any Tylenol? Like those sorts of things that we'll ask you. So it's a way to, and there's a lot of folks that have enrolled in this voluntarily, and it's a way for them to actively monitor how people are responding to this vaccine. There are two other systems as well. Vaccine Safety Data Link is one where they do a detailed data collection, but it's only from a subset of healthcare providers that are situated across the US. And then there's one called the Clinical Immunization Safety Assessment Project. And that's where any healthcare provider can reach out and say, I have something that might be a reaction to a vaccine. Can we take a look at that? And they'll consult with them. So when you have a vaccine that's under emergency use authorization, any healthcare provider is obligated to report certain events with this. And so if there's any administration errors, any serious adverse events, and then for this in particular, they want to know if there's any cases of MIS-C, which is one of those complications that can happen from COVID-19 infection. And then of course, if folks do get COVID-19, that results in hospitalization or death after the vaccination, they want to know about that as well because they want to continue to monitor efficacy. So what have they found? So they just released in the Morbidity and Mortality Weekly Report a few days ago the first set of data, and this was based on the first 13,700,000 doses that were administered. And it was more of the Pfizer vaccine because that was out a little bit earlier than the Moderna vaccine. And so I'm showing the Pfizer data here, but the Moderna was actually very similar. They just only had the first dose data for that. What they found was that the side effects were, pretty much what we expect from a vaccine. And I have to say that as adults, we get a little bit, I think sometimes we forget what typical vaccine side effects are, except for if we think about what our kids went through when they got their vaccines. And so if you have kids and you remember when they got their childhood vaccines, they probably got a fever and were pretty cranky for a couple of days. And that's maybe how you're going to feel after you get this vaccine. So if we look at our typical side effects here, they're things that we would associate with an immune response. So injection site pain, that's this dark blue line. So you can see after the first and the second vaccine, this is the percentage of people that reported it. Lots of folks had a sore arm after they got this injection. That's pretty normal because inflammation is a normal response when you're having any sort of immune response, and that's going to make your arm pretty sore. So that's to be expected, but that was really common and maybe more so than with some other vaccines. Other common things were fatigue, headache, and then muscle aches and chills. All right, so fatigue, headache, muscle aches, and chills. And you can see, like, this is our first vaccine. This is one day after the first vaccine, and then this is one day after the second vaccine. So what has been found, and I know quite a few folks that have had this vaccine now, is that usually you feel a little worse after the second vaccine. And so just be prepared for that. As far as serious events, so they did, looking at all these doses, looking through VAERS, there were 119 deaths that were reported. And of course, these are any temporally associated deaths. They have not finished doing their background checks on all of these. The ones that they have not found anything that would indicate it was from the vaccine. Most of them were in long-term care facility residents, and they expected, with that number of doses of vaccine that were administered in the long-term care facilities, they expected about 7,000 coincidentally temporally associated deaths. So really nothing surprising there. It doesn't appear that there was any increase in deaths with use of the vaccine. Looking at serious adverse effects, there have been some reported. Again, they have not finished their investigation on that. But if we looked at the clinical trials for the Moderna and the Pfizer vaccines, with Moderna, they didn't really find any severe adverse effects that were linked to the vaccine. The number of severe adverse effects that were reported was the same in the placebo and vaccine groups for that trial. For Pfizer, it was similar with the number of effects, but they did find four individuals out of the about 40,000 that were in the trial that did have some adverse effects that were linked to the vaccine. One was a shoulder injury that had to do with the way that the vaccine was administered. There was one that was severe lymph node swelling in the armpits, to try to put it in layman's terms. And then there was one proximal ventricular arrhythmia that was concerning. And then there was one that was, it's basically a tingling sensation in the leg that was associated with this. So they did have a couple of adverse events that may have been linked to the vaccine in the trial for the Pfizer vaccine, but those were just a few out of many, many people that got the vaccine. And I would add that many FDA approved vaccines have a lot more potential adverse effects than those. All right, allergic reactions have gotten a lot of press, and so I do want to just very quickly go through that. They're expected for all vaccines. You get about 1.31 cases per million vaccine doses on average. The data from this first month indicate that that's slightly higher for the Moderna and Pfizer vaccines. There were 62 anaphylaxis cases out of the 13 million or so vaccines that were given, making it about 4.5 cases per million vaccine doses. And that is a little bit higher than average, but it is lower than some other FDA-approved vaccines, such as the live attenuated herpes zoster vaccine has a rate of 9.5 cases of anaphylaxis per million. So what it means is that you do need to be cautious if you have a history of allergies, especially if you've had an allergic reaction to a vaccine. Review that ingredient list with your doctor. And be sure that you remain at the medical facility for 15 minutes. This is typically advised for any vaccine, but I think folks sometimes get lax about it. But don't run off right away. Make sure that you're going to be okay before you leave. If you have a history of anaphylaxis for any reason, even if it was like, an allergy to peanuts or something, they do want you to wait 30 minutes, and especially if you've had an allergic reaction to any vaccine. So just make sure you don't leave the facility right away after the vaccine. All right, efficacy. So you may have already heard about efficacy for these, but very quickly, the Moderna vaccine is amazingly effective. I don't know how else to put this. Like, this efficacy just blew me away when these results came out. 94% effective. And they did test this across a bunch of different age groups, and it was basically the same in all the age groups, from their oldest age groups, which were up into the 70s, down to their lowest age groups, which these did stop at between 16 and 18, depending on we're talking about Moderna or Pfizer. But they worked across all the age groups very well. Pfizer, and again, what you're looking at here, you've got your vaccine versus placebo, the number of cases, all right? So you can see with the vaccine, it just really just stopped those cases, efficacy of 95% for Pfizer. And then the new vaccine that's going up tomorrow, you can't really compare the data. It's not exactly the same. These trials took place in a couple different places. And so in the US, it was found to be 72% effective, Latin America, 66%, and South America, 57%, which does not look all that fantastic compared to Pfizer and Moderna. But for an average vaccine, that's pretty good, actually. There were some differences in the trials, though. And so the Moderna and Pfizer trials were mostly in the US. Moderna was exclusively in the US. And they were really before some of these new variants came out, whereas the Johnson & Johnson trial was more international. And so it was up against some of these newer variants. And so that makes a difference. And they also measured different endpoints. So the endpoint for Moderna and Pfizer, they were looking at just symptomatic disease, whereas the Johnson & Johnson was looking at moderate to severe disease. So not exactly apples to apples if you try to look at these. Now, someone had asked, can I get COVID if I'm fully vaccinated? And yes, there is that possibility. And so that's where you'll notice these are not 100%, right? And so there is a possibility that you could still get COVID if you're fully vaccinated. Even, if you think about folks who get infected naturally with anything, some folks develop immunity and some folks don't. So there's no one thing that's going to make everyone immune. So you do still have some risk, but that risk is reduced. All right, so this race between vaccines and variants. So every infected individual provides opportunities for SARS-CoV-2 to mutate. And so the more people that are infected, the more different mutants will develop. And we've got a lot of people that have been infected. And so therefore, there have been some mutations. We are fortunate that SARS-CoV-2 does not mutate as fast as some other viruses. So it doesn't mutate as fast as influenza. It's about half as fast as that. And HIV, it's about a quarter as fast as HIV. So it's not a super fast mutator, but it does mutate. And so the mutations we're concerned about are the ones that change that S protein, because that's the target. And this right here is the S protein. That's what we're targeting with the vaccine. So the first mutation that showed up was this D614G. And that was actually so prevalent that all the vaccines work against it, because that's really what the vaccines were tested against. That was present in the communities across the US before these vaccines were being tested. Now, there are a few new strains, and these strains each contain several mutations. So the B1.1.7, which you may have heard of as the UK strain, this strain has been tested. There have been several small preliminary lab studies looking at how well the vaccine works against this. And one of those studies out of the three indicated that there might have been a slightly reduced antibody efficacy in neutralizing this strain. That was for the Pfizer vaccine, but it was only slight, and we're not sure what the clinical significance of that is, because these are just lab studies. For the South Africa strain, again, there have been two small preliminary lab studies that indicated slightly reduced efficacy of both the Moderna and Pfizer vaccines. And so we do think that there might be an issue with that. Not that it's not going to offer any protection, but that it will not be quite as protective against that strain. And then the P1 strain, or the Brazil strain, is another new one that has come out. It shares a lot of mutations with the B1351, the South Africa strain. So we think it will be similar, but there really are not any good studies on that yet. Now, so I had one question that asked, what's the difference between a variant and a mutation? So in the news, they're probably using them interchangeably. A variant typically refers to a strain, and that strain may contain many mutations, whereas a mutation refers to one specific little change in the DNA. But I suspect that when they talk on the news, they may be using those two words interchangeably. So some things to keep in mind about this: the lab studies only look at the antibody response, and T cell responses play a role with this as well. So it is possible that although the antibody response doesn't work great, that the T cell response may still work okay. So there's all sorts of reason to have hope that this is still going to be okay with these vaccines. And that reduced antibody response that's seen in the lab may still offer full clinical protection, but we don't know that yet. Certainly, the results from the Johnson & Johnson study make us wonder, because it was less effective in the areas where some of these variants are coming out. But in good news, Moderna is actually already working on a booster that's specific to the B.1.351, thinking that maybe we're going to need to have booster shots. And so I had a question: will this be like the flu, where we have to get a vaccine every year? I hope not. If we can get the infection numbers down enough, it mutates more slowly. So hopefully we won't have to have a shot every year trying to play catch up every year. But if we continue to have high infection numbers, then that is a possibility. My thought is more that it would be maybe we're going to need every couple of years until we get this under control. And then hopefully we won't be needing to do that anymore. We'll just have one shot. And I do want to note that here in Blount County, I feel like we're making some progress on that. About 10% of Blount Countyans have had COVID, had been diagnosed with COVID. And as of this morning, about 12% have been vaccinated with at least their first dose of vaccine. And so there's some overlap between those two groups, but there's also probably some amount of underestimation of the folks that have been infected. And so we're probably sitting at about 25% of Blount Countyans have some immunity right now. And so that's reason for hope, I think, that we're moving in the right direction. It certainly isn't herd immunity. If you've ever heard that term, we probably need to get to about 80% of people immune to get to where this virus can't spread well in the community, but at least we're making some progress. All right, I've really talked for too long. All right, very quickly, what to expect when you get vaccinated. The injection doesn't usually hurt more than any other vaccine, but do plan to wait at the facility afterwards to make sure you're not going to have a reaction. Expect your arm to hurt for a couple days, and do expect to have some side effects, particularly if you've had COVID with that first vaccine. You'll probably have some side effects: fatigue, headache, maybe a fever. So expect to have that, or if you haven't had COVID, probably with the second vaccine, you might experience that. So just don't make any big plans for the next day after your vaccine. And know that immunity is not instant. It is going to take about 10 days to start to develop immunity after that first shot, and you won't really have full immunity until about 2 weeks after your second dose. And it is important to continue to wear a mask in public, because we do not know if these vaccines prevent asymptomatic infection. That has not been tested. We know that it prevents symptomatic infection, but not asymptomatic infection. So we don't know if you could still carry the virus and give it to others once you've been vaccinated. So how do I get vaccinated? If you want to get vaccinated, there is this great vaccine finder that's on the Tennessee.gov website, and that will help you to find locations where you can get vaccinated. Just make sure you check to see what phase that your county is in, or the county where you want to get vaccinated. You don't have to go to your own county. And please do be patient with our Blount County Health Department. I have some friends that work there, and I understand that some folks have gotten very testy lately and kind of angry because they don't have enough vaccine for everyone right now. But do know that they are trying to get the vaccine out to folks just as quickly as they can. All right, those are some resources I had, and then some questions that I had received in advance. I had one person ask if I had a family member that couldn't receive a vaccine for medical reasons, can they get the COVID vaccine? And the vaccine that the person couldn't receive was one that's very different from the COVID vaccine, but that's still something you would really need to discuss with your healthcare provider to find out exactly why it was that they didn't want them to have that original vaccine. This was an interesting question. Could the DNA wake up sleeping cancer cells causing cancer? The short answer is no. Of course, the messenger RNA doesn't have any DNA that's involved. So that's certainly not going to have any impact on the cells at all, really, for long term. And then the adenovirus, I can see where this question came from because some of the early adenovirus vectors could potentially cause cancer, but they have made these now replication deficient. Ad26 does not incorporate into your DNA, and so they're not going to be likely associated with causing cancer. How do you respond when a high-profile doctor or scientist says the vaccine is unsafe? So I guess what I always like to do is look for scientific consensus. And so if I'm looking for information about any medical topic, I want to look to the American Medical Association or the American Academy of Pediatrics, and not to just one person. When one person says something, there's a lot of different reasons that can happen, right? I mean, sometimes people are just looking for their 5 minutes of fame, or other times you'll find that they're trying to sell you things. So I tend to put less impact into one person and more impact into, what are groups saying about this vaccine? Oh, this was a good one. Are fetal cells used in the production of COVID vaccines? And so when you do scientific research, there is a lot of use of different types of cell lines, and quite a few of those cell lines are derived from fetal cells. And so for the Moderna and Pfizer vaccines, there were some fetal cell lines used in the original development, like the R&D part, but they're not used for production. For the adenovirus vectored vaccines, those are grown in fetal cell lines. And so, and that's not unique to those. There are quite a few of FDA approved vaccines that are also grown in fetal cell lines. So the rubella, chickenpox, Hep A, and shingles, those are all grown in fetal cell lines as well. So it's not an unusual thing, but if that's something of concern to you, then you probably would want to choose the Moderna or the Pfizer vaccine. How long will immunity from the current vaccines last? It would be nice to know. So far, they have studied 4 months out and have found that the immunity is largely persisting as far as the amount of antibody being produced. It drops a little bit, but that's expected at 4 months. So we don't know. That's something that we're just going to have to wait and see. It's the same as for natural immunity. We just don't know yet. Are different vaccines better for different populations? So I did some research on that to answer this question well, and it turns out that no, they all seem to work equally well in all the age groups that were tested. The way that they broke up age groups were different from vaccine to vaccine, but all of them did test from 18 to 55-ish. And then they also all had groups that were like from 55-ish to 70-ish, and then groups over 70. And it worked the same for the 3 vaccines that we've focused on tonight. It worked the same in all those groups. So there's no one vaccine that's better than the other for any population. And then this one was, given the choice, which vaccine would I want for myself? I would want the one that I can get in my arm now. Honestly, I would really like to get a vaccine. I suppose if by some miracle someone came over and offered me my choice, I would take Pfizer or Moderna since they're more effective. But I'm not going to wait. I'll take whatever I can get first. I think that it's important just to get vaccines in as many arms as possible to try to cut back on the spread, so we cut back on the development of these new variants. All right, so now I'm going to ask my colleague, Dr. Siopsis, if she can share any questions that have shown up in the chat. And I apologize for the length.
We have lots of questions.
Okay.
But before I do that, I have to do a thing. And that is that I am going to put a link in the chat. Part of this this whole thing that we're being able to do is funded by federal funding from the National Science Foundation, and we are collecting data about participants who have listened to this. And so I'm going to put a link here for those of you who are willing to go ahead later at the end, not now while we're talking, click on this and give us some feedback about this if you're willing. We would totally appreciate it. It's part of the reporting to the NSF that we can do, and they have given Maryville College lots of funding, so we want to thank them by providing them with the data that they want. Now, getting to questions. There's a question from Shannon Milliken asking if there are indicators to the entities distributing and administering vaccines if the mRNA in the vaccine has degraded, i.e., are there any indicators that the doses haven't been handled correctly?
Okay, so not from just looking at the vials themselves, but they are required if they're storing these in anything other than the packaging that they're sent in, which has a very specific amount of time it can be kept. Then they are required to have recording systems for their freezers so that they know if that chain of cold has been broken. And so there's nothing on the vials themselves that would tell you that. So that is something that could happen. And that's one of the reasons why the Johnson & Johnson vaccine is so attractive. It doesn't require that. But they're certainly very strict about trying to monitor that things are kept at the right temperature. And you do have to have a 24-hour monitoring system on your freezer if you're going to be storing these vaccines.
I'm going to ask these in the order that they were asked. So they're going to jump around a little bit. One question from Dr. Klingensmith is, he would like to know, would the fact that some poor countries cannot vaccinate their whole populations pose a danger to populations of richer countries that have been vaccinated?
Absolutely. And that's why the World Health Organization is really trying to make sure that vaccines get distributed to many countries. There are lots of vaccines that have been authorized in other countries, and the World Health Organization has authorized several vaccines that we have not. And so there is a real push to get this vaccine out to everywhere, because if this virus is spreading anywhere, it's a threat to all of us.
Okay, here's another one. From Amy Howe, what does it mean by chimpanzee adenovirus or human adenovirus? What are the ingredients there?
Okay, so chimpanzee adenovirus is just okay, I should step back. Adenoviruses are viruses that typically cause colds or like diarrhea. So they're viruses that there's lots of them that we get as humans all the time. The chimpanzee one is one that chimpanzees get. We don't get it. So it's not a human virus. And the only ingredient is the virus. And so it's not going to replicate in your cells or anything. It's replication deficient. And then the adenovirus that you're using for the Johnson & Johnson vaccine, that Ad26, it's a human virus, but it's made so it can't replicate. And I should note, I forgot to mention this, that we have an adenovirus vaccine that's used in the military, and it's an oral vaccine, and it's actually not even attenuated. It's literally 2 live adenoviruses. So we certainly think that adenoviruses are safe when we're giving them live to our military recruits.
Okay, another question from Devet. Can you interchange the types of vaccine? In other words, can you take 1 dose of one and 1 of another?
Yeah, so at this point, we're going to say no. The testing has only been with, taking the Moderna followed by Moderna, or Pfizer followed by Pfizer. There's a lot of questions. Can you get by with just doing 1 dose? Can you take one now and take another one later? I don't think, if we're talking about a year later, there's probably not going to be a contradiction there. But as far as, if I got Pfizer for the first dose, can I get Moderna for the second dose? No, we don't have clinical data to support that.
Okay, so side effects of the vaccine indicate that the innate immune system is working. So somebody who's fully vaccinated had no side effects other than a sore arm and is asking, should they have had side effects? That person is saying that they are 75 plus. So they're a little worried, I think.
Okay. So in older adults, y'all are pretty lucky on this one. You were not lucky with the virus, but you're lucky with the vaccine because you don't have as many side effects. And so older adults generally don't have as strong of an innate immune response, just in general, don't have as strong of an immune response in general. However, with this vaccine, it's been working really well in that group. And so the lack of the side effects does not mean that it didn't work. And everyone's body is different. And so, we show how many people have side effects, but there's a whole lot there that didn't have side effects too, and it worked in them as well. So it doesn't mean that you don't have efficacy. It just means that you were fortunate that you didn't get the side effects. So it's not something to worry about.
Is it true that the Johnson & Johnson vaccine contains mercury, and is this a concern? Asks John Hattie.
That's a good question. I didn't look up all the ingredients of the Johnson & Johnson vaccine. However, certainly there have been other injectables that have contained small amounts of mercury, and that's generally not a concern. Sometimes that's used as in part of an adjuvant. So I'm not sure if that's in there. I didn't look up all the ingredients on that particular one, but if it is in there, it's not. It would be something that's been in other vaccines as well.
Overuse of it, from Kevin McNeil, overuse of antibiotics can result in resistant bacterial mutations. Does widespread deployment of the COVID-19 vaccine pose a similar risk with the virus?
So if we continue to have a lot of transmission of the virus, certainly when you have folks that are vaccinated and then could potentially be infected with a strain that evades the immune system, yeah, but that's the same concept if folks had been naturally infected with COVID as well. And so you certainly are creating a selective pressure for the development, or for the, I shouldn't say for the development, for the propagation of strains that avoid the immune protection that you have from the vaccine. But you get the same protection, the same sort of selective pressure if folks get naturally infected as well. And by reducing the number of folks who are susceptible to infection, if we can do that quickly enough, that's why I said it's a race between the vaccines and the variants. If we can get this vaccine deployed quickly enough, then hopefully we won't have all these new variants developing. But it is a race. It definitely is a race.
Okay, I actually, I'm waiting for a couple more people. If people have questions, put them in the chat, but I have two more questions. One of them is mine.
Okay.
You mentioned the VAERS.
Yes.
Which I've seen several people post the VAERS, in a variety of forums. And I'm wondering how they actually use that data, since, I mean, anybody can go in there and put anything, right? So I could go in there and say, without even being vaccinated, right? I could go in there and
Yes.
say, oh, I had this terrible. So
Yes.
how do they use that data?
So that data is a place for them to look for potential issues, but you can't take that data and just say, hey, there's all this horrible stuff happening, because absolutely it's like Facebook. Anybody can go on there and do anything. So the CDC, if anyone reports anything severe, like if you just say, oh, I had a headache, not a big deal. They're just going to assume you had a headache. But if you say, I had a heart problem or something, they are going to follow up with you. They're going to contact your doctor, the hospitals, everything else, and try to trace that back and figure out, okay, was this related to the vaccine or was this not related to the vaccine? Because they really do want to find if there's a problem. I mean, scientists and the CDC does not want there to be a problem, especially we have multiple options here, right? Like, it's not like we have to have one particular vaccine that works great. If there's a problem, we want to find it. So it's a way to alert that there might be a problem, but you can't just take that data and assume that it's all accurate.
Okay, and one final question from our dean, Dr. Klingensmith. Okay. If you're willing to speculate, oh, there's another question, sorry, but okay. This one from Dr. Klingensmith. If you are willing to speculate, is it reasonable to say that in some ways we were lucky with this pandemic because the virus so happens to be something we can use mRNA to address? So for other viruses, we can't necessarily expect a vaccine could be produced so quickly.
So that's an interesting question. It's not so much that this particular virus was special that we could use messenger RNA. Certainly, we could use this mRNA vaccine technology to address a lot of different types of viruses. But some viruses are really hard to prevent with a vaccine. So HIV is a great example. And I know early on folks were like, how come we couldn't come up with an HIV vaccine and we can come up with this one so fast? But HIV is a very different virus. It's a retrovirus. It attacks immune cells. It makes it way more complicated, the types of immunity that you need to produce for that. And so there are some viruses for which, creating any vaccine is problematic. And so a messenger RNA vaccine won't solve all our problems, but it is definitely a great tool that we have in our pocket. And now that we know it works, should we have another pandemic influenza outbreak or something like that, it's something that we could potentially use.
Okay, on the mutation question, we have a student asking, student Matthew Clark is asking, would you say that it is somewhat like survivorship bias when people focus on the vaccine causing mutation or evolution?
Oh, that's an interesting question. So that's a tough one to answer. I guess what I would say is that any therapeutic that you use is going to drive evolution of something that's going to evade their therapeutic. And so that doesn't mean you don't use the therapeutic. I mean, we still use antibiotics. And so it just means that you have to be aware of that and be looking for ways that you can improve in order to make sure that you don't get outcompeted, right? And so I think with the vaccine, it's not going to raise that risk of mutation and variants catching on any more than the natural infection does. But we also have to be aware that the more people that are vaccinated, the more hosts are only susceptible to the variants. And so we have to be ready to attack those variants as well. And so that's why we already have Moderna working on a new booster shot that would specifically attack that strain that's not easily fought off with the current vaccine.
I'm not seeing any more questions.
That's because I talked forever.
You did. Thank you so much, Dr. Brigati. Thank you so much for sharing this expertise with us. It was really appreciated. I know by our students and I see some faculty and staff, and I see many community members. A couple people asked me in the chat if we're going to be recording, and we are recording. And we will be putting the recording up on our YouTube channel, which the Scott Science Scholars have their own YouTube channel. If you answer the survey, we will know your email address and we'll be able to share that link that way. If not, and that's okay, the same link, the same person you emailed to get the link to the Zoom is the same person you can email to get the link to the recording. I know some people would like to share this with family and friends who couldn't attend. So I appreciate everybody coming. I appreciate Dr. Brigati's time and expertise, and everyone else who, Lindsay Walton and some other folks on Maryville College campus that made this happen for us. So thank you.
Thank you all for joining us.